Okay, most of you probably know that your body is kept at a temperature of at about 37 degrees Celsius, about 37.5. I want to know why. Your body puts a lot of effort into this. I mean, if you're too hot, it will help you to sweat, which will help you to shed heat. You'll get flushed, which is because when blood goes closer to the surface of your skin, so you can lose more heat. If you get too cold, your body puts a lot of energy into warming you up. You'd start to shiver, which will produce heat in your muscles. You will go kind of pale as blood is directed away from the surface of your skin. And your hairs will stand up to trap a layer of air to kind of warm you up. Goosebumps are a leftover of where we used to have hair. So your body puts a lot of effort into maintaining an internal temperature of 37 degrees. Why? Well, the answer is pretty obvious from the title of this video. It's about enzymes. Enzymes work best at 37 degrees, or at least the ones in our body do, and they control chemical reactions that happen inside your cells, and in some cases, like in the digestive system, outside of your cells as well. They're not just found in us. If it's alive, it's going to have enzymes. They're essential for life. Enzymes are proteins, and they're made up from amino acids which are joined together. They are extremely complex, and for a molecule, they're pretty big. We often use a lock and key analogy to describe how enzymes work. In this case, the enzyme would be the key, and the lock that it fits into is the substrate. That's the target molecule that it's going to work on. Each enzyme is very specific to a certain substrate and will only work on that substrate. Okay, here's a little example. We've got a substrate. Part of the enzyme that binds on to the substrate is called the active site, and it's very specially shaped to fit into the substrate, just like a lock into a key. Okay, so the enzyme is very specially shaped and will bind onto the substrate. And then it will do something. Now I say something because some enzymes will chop the substrate in two. Others will join two substrates together. There's loads of different ways that enzymes can work. In this case, this example is a digestive enzyme that's going to break the substrate down into two pieces. It binds onto it at the active site, and in this case it changes it into two, change the colour to make it a little easier to get your head around. And the two bits then. Now the key thing to note here is that the enzyme hasn't really changed. The substrate molecule that it hit was changed, but the enzyme itself hasn't. Instead, it's ready to work again. And it will keep working. As long as there's enough substrate, it will just keep ticking along. Okay. In general, in a chemical reaction, if you heat it up, it will happen faster. If you increase the temperature, the rate of the reaction will increase. This is because when you heat the little particles, the little molecules, they will bounce around more, they'll have more energy, which means they're more likely to hit each other, and they're more likely to hit each other with enough energy to react together. From a chemistry, this is the activation energy in a reaction. So let's have a look at a graph of our enzyme and our substrate. So at first, here we've got on the x-axis, we've got the temperature in arbitrary units, means they've just made it up, should really be degrees or whatever. And on the y-axis, we've got enzyme activity, so how well the enzyme's working. And as you can see, as the temperature increases, I'm just going to describe the graph first, then we're going to look at explaining it, just like we did in the practice ones. At first, as the temperature increases, the enzyme activity increases. But after a certain point, even if you keep increasing the temperature, the enzyme activity starts to level off. At its optimum, at the optimum temperature, the enzyme is working the fastest that it can. If you keep increasing past this point, the enzyme rate actually drops off very, very suddenly. Can you think of why? Well, the reason why the enzyme rate drops off so quickly has to do with the shape of the enzyme, like we were talking about earlier on. Here's a picture of our enzyme again, with the active site, the kind of mouthy bit there on the right hand side. So if you heat something, in addition to moving it around, you can also change its shape. Remember that these enzymes are molecules made up of atoms, and the atoms are bonded to each other. Now obviously if you heat that molecule, it's going to change shape. Some atoms will change and bind onto something else, and overall the shape of the molecule will change. Remember now that the enzyme is extremely specific to its substrate, that lock and key analogy again. It will only fit provided the active site literally fits it. If the active site changes shape from being heated, it won't be able to bond onto the substrate anymore because it won't fit anymore. It'll change from that to having an extra sticky outy bit, or to having a different shape. If this happens, the enzyme can't do its nature anymore, so we say that it is denatured. It is not dead because it was never alive. It's a molecule that was never alive in the first place, so never say it was dead, it's been killed or anything. It's just being denatured. It can't do its nature anymore. Okay, here's a picture of our enzyme again. We've talked about how temperature can affect enzyme action. We're going to have, when we talk about something's pH, we're referring to how many hydroxide ions it's got or how many hydrogen ions it has. The more acidic something is, the more hydrogen ions it has, and the more alkali it is, the more hydroxide ions it is. So in this case here, we could go either way, but let's say that the green dots represent hydrogen ions. So at a certain pH, there will be a certain amount of them, a certain concentration, and the enzyme is used to working in those conditions. Its shape will be, it'll be the right shape under those conditions with that amount of ions in. If you change that by taking away a lot of these ions, or even by adding more of them, you'll change the shape of the enzyme again. If you notice, the active site has changed shape again. It will no longer be able to bind onto its substrate, and once again it's become denatured and won't fit onto the substrate anymore. Okay, if we were to graph that, again we'd see that there's an optimum pH at which the enzyme activity works the best, or the rate of enzyme action works the best. In this case, it's a pH of 8. Now, different enzymes have different optimum pHs, just like different enzymes have different optimum temperatures. Alright, well done. Here's a picture of a little puppy high-fiving a little kid.